After mutations, we have a most important topic, Lac Operon. Lac Operon is actually a messenger RNA, and in this messenger RNA, it is actually a polycisternic messenger RNA, and it's belong to prokaryote E. coli. We have known that E. coli are one of the lactose fermenters. So how E. coli became a lactose fermenter, we will discuss here. So in the E. coli messenger RNA, we have a lac I, we have a promoter, we have cap binding side, we have the promoter side, we have the operator side, then we have the lac A, lac Y, lac Z. So E. coli preferred metabolic substances glucose, but it can sustain by using lactose via this Lac Operon mechanism or Lac Operon metabolic. So E. coli has a particular type of mechanism in which E. coli can sustain without having glucose or in a glucose deficient media. So whenever there is an environmental change, like whenever there is low glucose or lactose unavailable or lactose available, so environmental change can lead to genetic modulation or genetic activation or genetic change. So first describe these four important genes. So lac I is actually encodes the regressor protein. This regressor protein is actually a negative regulator, and it loves operator or operator site. It loves operator. It binds to the operator in order to negatively regulate this lac operon messenger RNA, negatively regulate the transcription of these enzymes to make the E. coli sustainable in a glucose deficient and lactose sufficient environment. Then we have the lac Z, and this Z produces this gene produces an enzyme known as lactase enzyme, which converts lactose into glucose. Then we have the lac Y, and this lac Y make permease, which make the lactose to enter into the cell. So these are the general descriptions of these genes. So how this lac operon operate? Whenever there is a low glucose and there is high lactose available, usually E. coli uses glucose and survive. But whenever there is a glucose deficient environment, the E. coli switch its metabolic function by switching on the messenger RNA and producing particular enzyme which catalyze those lactose molecules. So whenever there is a high lactose, so there is a high transcription of these enzymes to metabolize the lactose molecule. So the negative regulator, it there is an enzyme known as allosterase, which is the inducer, which bind. This is the first step, this is the second step. Allosterate inducer, it actually bind and inactivate the repressor molecule. If the repressor is bind to allosterase, or if the repressor is inhibited from going to bind the site of operator, so allosterase bind and inactivate, bind to repressor, inactivate the repressor, so there is no binding to repressor, binding to operator site. So now we can open the gates for the polymerase to act and translate these Lac Z, Y, and A gene to form different types of enzyme which help to metabolize the lactose molecule. So now no operator binding, so operator is open. So low glucose, it activates, second step is low glucose activate adenyl cyclase, which releases a lot of cyclic AMP. This AMP goes induce or goes and bind to the cap area on the gene activator protein, and this activator protein now activate the polymerase enzyme. So over here camp and cap, they now start transcription, or they are the positive regulator of this Lac Operon. So now they are activated, so polymerase come and bind to the polymerase site, gates are open, operator gates are open. So now the translation begins, and Lac Z is translated into lactase and permease, and now the bacteria can metabolize lactase molecule in a glucose deficient medium and lactase high medium. So strongly transcribe or strongly express these genes. So this is the general mechanism how Lac Operon works. So now there are some conditions in which there is first one is physiological. Physiological means high glucose and no lactose. So physiologically what happened, high glucose, the Lac Operon is inhibited. So no lactose mean there is no transcription of Lac genes. So the repressor, the regulator, negative regulator, repressor is bind to the operator and close the gates for polymerase. So no transcription of the lactase and permease enzyme. So Lac Operon is not activated. The next case is when there is high glucose and high lactose, or lactose is available, then repressor very low or basal expression of these enzyme, like into 5% of these enzyme Lac Operon activation, or there is 95% of glucose metabolism. So very low or basal activity of the Lac Operon happen. So now when there is a low glucose and low lactose, so what happened here? No lactose, it means no transcription. So repressor again bind to operator. So no transcription of the lac gene, because as I have mentioned this, that first and most important part is we have to remove that watchman repressor from the gate operator in order to proceed further. I have first discussed the high lactose environment, then I discussed the low glucose. So high lactose is very much important in order to operate this lac operon, because if you have low glucose and you don't have any lactose, then the repressor already bind to the operator. So this case is very much important, that we have second condition, we have satisfied the second condition, but we haven't satisfied the first condition. So whenever there is low glucose and high lactose, then this mechanism happen. So these are the three important conditions, and these are the important MCQs over here, like lac regulator, and also there is allosterase binding, then who is the strong positive regulator, who is the negative regulator. So these are the kind of MCQs pop up in the board exam for this lac operon genetics.